Hwilu from Dr. Sabine Kuss research group in the Department of Chemistry. Carboplatin is listed by the WHO as an essential medicine to treat various human cancers, including ovarian cancers. However, 75% of patients relapse within 18 months. Of these patients, 85% experience drug resistance. Worse, the underlying mechanisms of resistance is poorly understood. Therefore, there is a need for a tool to detect and study drug resistance in cancer. Electrochemistry is found to be suitable for our purposes. In our project, first I electrochemically characterize carboplatin, then I detect and compare the carboplatin consumptions between drug-susceptible and drug-resistant cells. Our hypothesis is that susceptible cells consume more drugs than resistant cells if uptake inhibition is responsible for drug resistance phenotype in those cells. Figure 8 shows the cyclic photometer in which we vary the electric potential to measure the electric current as our signal. The chemical structure you see is carboplatin. This figure shows that we can detect carboplatin by taking its electrons at 0.76 volt in an oxidation reaction representing in this peak over here. To determine if we can detect carboplatin in a variety of samples, from wastewater to blood in our body, we need to change the pH and see if we can still detect it. Indeed, we can detect carboplatin from pH 1.5 to pH 9. Next, we want to see if we can detect carboplatin in low concentration in our body. Therefore, we lower the concentration of carboplatin, and as a result, the peak current decreases. This figure shows that we can detect carboplatin concentration as low as 30 micromolar. Figure DI shows the drug-susceptible cells. Figure DII shows the drug-resistant cells. To detect drug consumptions, we expose different cells concentration to carboplatin solutions. Then we remove the cells and measure the carboplatin concentration in the remaining solutions. As the cell concentration increases, the peak current decreases, indicating a drop in carboplatin concentration, and thus drug consumption is detected. Finally, we want to compare the drug consumptions in susceptible and resistant cells. These two dotted and solid black lines show that carboplatin does not degrade after our incubation period. The red and blue line shows that no significant difference in drug consumption between susceptible and resistant cells are detected. With that, we conclude that the decrease of drug consumption is not the only mechanism for drug resistance. Thank you.